All right, so thank you, Simon. And I'm going to also thank the organizers. I guess Mohammed and his team always done an amazing job, and it's just actually first time in over 2 years I'm actually standing on a podium in a room with colleagues and friends, which feels good. All right, we've heard a lot yesterday and this morning about nucleic acids as drugs. We've learned about how the challenges to try to get them into cells so that they can actually meet the target. So we're going to change a little bit of topic in here. And I guess for my talk and the follow-up talk with with Lucia, we're going to try to see that oligonucleotides can also be used as biomarkers for diagnostics. They're not just targets for drugs. They can also be used for new point of care diagnostics. So this is what we've been doing in my lab for quite a few years since I've moved to Imperial College, about over 12 years ago now, trying to develop and engineer some platform technologies for detecting nucleic acid biomarkers, focusing primarily in bodily fluids. So basically trying to avoid all of the challenges. We know it's hard to go into cells, so let's try to go for cell-free nucleic acids and trying to make our life as easy as possible. So on this slide, I've just shown the three main platform technologies we're working on. We're working in lateral flow assays. Lucia is going to talk about her work focusing on that, so I'm not going to mention it at all. We're also working with nanopore technology. Always good to mention nanopore when you come to Oxford. Always a good way to have the support of a crowd. And the topic I'm going to focus on today is the topic of microneedle patches. We're using this platform technology for a range of different applications that goes from cancer diagnostics to prenatal testing. Seems very, very different in terms of engineering and platform technologies, but pretty much the same. And for today's talk, as I said, I'm going to focus on our work developing microneedle patches for point of care detection of skin cancer. What we're trying to do is have a new diagnostic test, which will remove any surgical intervention. So no cutting your skin, and try to have something which is very quick so that you don't have to wait for weeks or months to actually get a proper diagnostics. So why focusing on skin cancer, you may ask? Well, if you look at detection early, skin cancer is not known to be one of the deadliest cancer. There are some much worse ones, but which is true for melanoma, for example, one of the deadliest forms of skin cancer is similar to what is true for cervical cancer, breast, or colon. If you have a high survival rate, if they are detected early, this prognosis drops dramatically if cancer is detected very late. Skin cancer, like many other cancers, is being diagnosed when you actually suspect something is wrong with you. You have a lesion that looks a little bit touchy. You then go and see your GP or pharmacist, have a second opinion, and then they're going to send you to then see a dermatologist and so on. So multi-step process, you have to see multiple healthcare professionals before you can actually end up with a diagnosis. And again, this relies on you being confident and wanting to go and see a GP to actually be checked. So skin cancer remains, and something people not many know, we talk a lot about breast cancer for women, prostate cancer for men, where skin cancer is has no gender bias. It affects both men and women, and is actually the most common form of cancer in the UK and keeps increasing. Incidence increases between 1% and 2% every single year. So you've got a range of different forms of skin cancer. The deadliest one are cutaneous melanoma or Merkel cell carcinoma. As I said, deadliest with over 60,000 deaths worldwide every single year, and 16,000 new cases diagnosed every year in the UK alone. So those are normally the ones that scare people the most, the one you want to be sure you detect early. There are other forms of skin cancer, which may seem more benign in some ways, still potentially dangerous. Basal cell carcinoma, BCC, or cutaneous squamous cell carcinoma, CSCC, as I say, more common, maybe less deadly or may take longer to kill you, but actually very hard to diagnose, especially at a very early stage. So how do you actually diagnose cancer, skin cancer? I've touched on that already in the first few slides. Well, it normally starts with a very, very subjective and inaccurate process which is a simple visual inspection. That means if you go and see a trained professional, whether it's a pharmacist or a GP, even a dermatologist, the first thing they will do is have a close look at your skin lesion or your mole, and then look at kind of specific characteristics, asymmetry, borders, the color, the diameter, and whether it actually kind of changed shape over time if you've been to the same kind of dermatologist or GP or multiple times to get something checked. So obviously that's a massive part for the dermatologist or GP to kind of decide whether this is something that could potentially kill you or something you can easily live with. That means in many cases, if they have any doubt that this could potentially be cancerous, then they will normally refer you for a biopsy. That means that a lot of biopsies are basically being conducted every year. Only 6% of these biopsies actually turn out to be malignant. That means we're going to remove a mole from your skin, leaving a scar, maybe fine if it's on your back or on your arm or on your legs, a bit less fine if it's on your face. And this is basically, a lot of those are necessary, costly, and can sometimes in themselves be tumorogenic. Every time you start cutting a bit of skin, you can actually kind of make something more dangerous than it was initially. This is also very costly for the NHS, that spends over £35 million every year on unnecessary diagnostic procedures just for skin cancers. As also mentioned before, time to result is pretty long. You will very rarely have a biopsy done the same day you actually have your first diagnosis. You will need to have a follow-up appointment, then have a biopsy taken, and then sample being sent away for analysis, which is again going to take a few weeks before you can get the result. So this is long and very distressing for the patient, the fear and the unknown, not knowing what's going to kind of come back from the labs. So as I said, skin biopsy remain the kind of a gold standard for skin cancer diagnosis. You've got 3 different types of kind of biopsies. They either kind of cut the entire mole and kind of then kind of a few kind of suturing to kind of close the gap. You can have some shaving, which is basically kind of cutting the mole, or just kind of punch biopsies, all of these depending on the size of the mole you actually want to analyze. So that remains the gold standard, the most specific and the best way for a dermatologist to tell you, yes, it is cancerous, or no, it isn't. Obviously, lots of research is being done in this area to try to kind of come up with technologies which are less invasive and equally sensitive and specific. So starting from the right, a lot of kind of image-based kind of artificial intelligence kind of a technology, basically improving the way the image of your mole is taken to try to look at these kind of features that can actually be characteristic of a cancer. Yes, it is much more accurate than just a simple subjective visual inspection. Less subjective, you trust the software to actually kind of analyze the image, but the sensitivity is still fairly limited. You may say 80–85% is not that bad, but that still means that you may have 15 to 20% of cases where you're going to be told, yes, you're fine, go home, while you actually have a cancer that may kill you a few years later. The kind of one of the most interesting new technologies that comes from the US is a kind of company called DermTech, and uses, you can't really see very well there, but this kind of translucent kind of patch is just a sticky patch. If you want like a plaster, you put directly on your mole, then kind of pull it off. That's going to kind of capture some of the cells which are on the mole, and then they send it to a centralized lab that are going to detect a range of different molecular biomarkers. So that's one of the rare few kind of diagnostic tests which is based on molecular biomarker rather than visual inspection, and gives you a pretty good level of sensitivity and specificity. It's still not point of care. You have to wait for the samples to be processed and analyzed in the lab. And it's something which is extremely costly. We're talking there almost kind of $500 if I remember well. So not something you can do as a routine test if you want. So what is our vision in this area? So trying to find something that kind of improves the way skin cancer is being diagnosed. So that's something we kind of try to kind of name MPatch, which would be a first point of care device that could simplify and shorten the process of skin cancer diagnosis. I've mentioned melanoma, but basically any skin cancer will have to be removed in the end. So we want a test that can tell you, is it cancerous and has to be removed, or is it just benign and you can carry on living? And this is work which we had initiated with MIT, and I may disclose that this has been published, part of it, and also patented, but I think that's just kind of present still the results. So the idea is the following. You have this kind of microneedle patch, which you would apply in a dermatologist or a GP's kind of office, wait for a few minutes, and you can directly get a result based on the detection of molecular biomarker from your device. Idea being something which is pain-free and non-invasive. I'll talk about the technology itself a little bit later. As I said, could be applied by the GP and dermatologist alongside visual inspection and applied near the skin lesion, not on the lesion itself, but just nearby. And what we're trying to do in here, where we're kind of referring back to nucleic acids, we're trying to have an in situ unpatched electrochemical detection of a panel of cancer-specific biomarkers using bespoke molecular probes. All right. So idea there, as I've kind of touched on, we try to look at biomarkers from liquid biopsy. So liquid biopsy, massive improvement from kind of removing a mole or actually doing punch biopsy. Less invasive. It gives you a more kind of minimally non-invasive diagnostics, a more comprehensive tissue profile, and it's much faster and safer than a tissue biopsy. There are some pitfalls. If you look at blood, for example, which is the most interrogated bodily fluid, before you want to analyze biomarker from blood, you require heavy sample processing. You can't do PCR, for example, on a blood sample without extracting the RNA beforehand. And the tumor-derived biomarkers tend to be highly diluted. It's in systemic circulation. If you want to kind of take blood from your fingerprinting, for example, and your mole is on your back, biomarkers coming from the mole will actually be diluted with lots of other biomarkers coming from elsewhere in the body. So then you have to think, is there any other type of bodily fluids which may be more appropriate for kind of skin cancer diagnosis? And obviously, skin fluid, you try to think immediately about skin interstitial fluid. So this is just kind of for those not very familiar, as a kind of a schematic of the different layers present in skin. You got the epidermis there, and then the kind of in pale pink, the dermis, and then going a bit deeper into the tissue over there. The skin interstitial fluid is normally located just at the interface between the epidermis and the dermis. So that's just the fluid, but just connecting all of the kind of cells over there. In red, you've got here the kind of blood capillaries, which are in the dermis. So if you adjust where you sample your fluid, you'll be able to have only skin ISF and not blood, which is exactly what we want in this case. And the kind of small kind of nerve here represented in blue. So if your patch doesn't go too deep into your skin, it's going to be totally painless. You're not actually reaching the nervous system. So pain-free and kind of blood-free kind of a skin interstitial fluid. All right, then sounds nice in terms of practicality, but what kind of biomarkers can you actually find in skin interstitial fluid? So that's a pretty new field of research, and there's not a massive amount of kind of data compared to kind of biomarkers present in blood plasma, for example. But recently it's been shown that skin ISF contains exactly the same RNA biomarkers as kind of blood, for example, microRNA, which are our kind of biomarkers of interest, and also importantly, less junk than blood. When you want to do kind of analysis of kind of blood samples, for example, getting rid of red blood cells or massive proteins is an issue. You don't have this problem when you try to detect biomarkers from skin ISF. Also importantly, you can sample the skin ISF near the site of the tumor, where the concentration of tumor-derived biomarkers are going to be much higher, much less diluted than in systemic circulation. As I said, what we want to work on is circulating cell-free nucleic acids, short non-coding RNA, don't have to kind of introduce them in this audience. And we're going to start focusing on biomarkers which were already validated in blood and showed very good level of sensitivity and specificity for skin cancer. And we're going basically to try and see whether these biomarkers are also equally good in skin interstitial fluid. Just as an example, if you look at this combination of three microRNA, 155P, 149-3P, 193A3P, this kind of microRNA profile give you an area under the curve of 0.97 for the diagnosis of melanoma, which is as good as a tissue biopsy. All right, so we've got our biomarker of choice, we've got our fluid of choice. How do you actually kind of get to skin interstitial fluid and be able to analyze it. So there's not many techniques to do that. The most common one used in clinic is the one on top of the slide, the suction blister, basically kind of applying vacuum on your skin and trying to suck up whatever liquid comes off your skin. You have to kind of stay with that on your skin for about kind of, I think, 30 minutes or so. So not the most comfortable. From what kind of dermatologist says, patients hate that because it's very painful. Okay, it leaves no scarring, but it's very, very painful. And then you have these kind of blisters being formed, and then you can just kind of sample the fluid from these blisters. It gives you plenty of it. It's great because you can do plenty of studies on it, but patients really, really don't like it. So what we try to find is something which is much less kind of invasive or painful. Those are the type of microneedle. So it's very hard, they're so small, it's very hard to take a picture which you can actually kind of see the design on. This is just a patch there, and all of these small packs in here are basically microneedles. You can apply them on skin, and as you see, you will just kind of, they penetrate skin, but they will just leave some very, very small red marks on your skin, painless, and definitely kind of very fast as well. So microneedle technologies have been out there for quite some time now, and they're expanding quite a lot, but they've mostly been used for kind of drug delivery. So transdermal delivery of vaccines, for example, or any kind of drugs, you can have some kind of microneedle patch that dissolve when in the skin. You can load them with a cargo, apply them, and this is the main area of research, whether you look at kind of clinical trials or research paper. So anything in black shows application for delivery. Anything in green shows application for diagnosis. So you see pretty small in both cases. And clinical trials focus mostly on kind of cosmetics, then skin conditions, kind of influenza, diabetes, but very, very few, less than kind of 5% on cancer. All right, so what are we trying to do in here is trying to kind of combine all of this and try to kind of sample skin interstitial fluid using microneedle patches coated with a hydrogel layer. So this is a patch, hopefully you can see it a bit more there. The patch, for information, is about 1 centimeter square and has an array of 9 by 9 kind of needles on it. So this is the patch. It's made of a polymer, PLL, for those we're familiar with, and basically kind of melted under the oven under vacuum and then set, and that gives you this pretty hard patch. So this is the pre-coated version. We then use as a hydrogel alginate. So this is a natural kind of hydrogel polymer commonly found in kind of in algae, and you can also use in molecular cuisine for those who are actually kind of into this type of things, and kind of form actually kind of hydrogel by acylation of calcium ions. So you got this viscous solution, you put it in high calcium concentration, calcium kind of coordinate a range of carboxylic acid and OH groups and form a gel. So you first coat your patch with a layer of alginate, let it dry, still not cross-linked. So this is the second picture there. Then you will add the kind of calcium chloride, and then this is going to then cross-link the fibers of alginate, still bound onto your patch. This is an SEM image of a patch, of a needle, a single needle, before coating or after coating. The height of a needle is kind of 550 micron, but when you apply it onto skin, obviously not all of the needle goes into the skin, only 100 micron actually kind of go far within your skin. The hydrogel is dry, you apply it in skin, then hydrogel wants to rehydrate itself, loves water, and basically going to soak up the skin interstitial fluid in the skin. So this is a kind of an SEM image of a kind of a nice mesh structure of your hydrogel. And then you can see in here that's the swelling volume of your patch when you apply it into the fluid. That means our single patch will be able to kind of soak up or swallow kind of 6.5 microliter of liquid very, very quickly in less than 2 minutes, which is great for what you want to do. You apply it, get a reasonable amount of fluid in a very short amount of time. But what we want to do is not just sampling the skin ISF. We also want to detect microRNA biomarkers within the skin ISF, and ideally at the point of care directly on the patch. So we need to find a way to selectively capture and detect the microRNA within the patch. So to do that, we just kind of decided to modify our hydrogel with peptide nucleic acids. I noticed kind of always at this conference that they tend to be kind of less and less popular. People talk a lot about LNAs or kind of PMOs also out of fashion a bit, but PNAs in our hands work pretty well. So then we have to find a way of kind of attaching these PNAs onto the hydrogel. So this is the chemical structure of alginic acid, and we've basically tried to explore different chemistries. So this is ongoing work from one of the PhD students in my group, so Connor, and we've basically tried to kind of attach a range of different functional groups, whether they are DBCO, so constrained alkyne to the fibers. Chemistry works, but so hydrophobic that actually the hydrogel is impossible to redissolve after that. So you can't really do much with it, so not great. But you can then functionalize it with a normal kind of alkyne functionalized with a PEG group that still gives you the azide, sorry, not the alkyne, with a PEG group, that still gives you a nice soluble kind of hydrogel. We can also use maleimide thiol chemistry. That works as well. Thiol kind of a maleimide, but other way around, works well as well. Methacrylate groups also react very well for click chemistry with thiols. That works pretty well. Or a more kind of unusual chemistry, opening one of the ring of the alginate through oxidation, giving you two aldehydes, which then allow you to kind of click some peptides on it. So this is just ongoing work of the chemistry to optimize it. The work we've kind of conducted afterwards is using this type of linker. So a PEG linker attached on the carboxylic acid of the alginic acid via kind of copper catalyzed click chemistry, also having a photoclavable linker between the PNA and the hydrogel. I will show you later why. And then done a bit of in vitro testing. So how does this work? Can we actually sample a specific kind of microRNA with these PNA hydrogel conjugate immobilized on microneedle patches? Experiment the following. This is your hydrogel on the patch. We add kind of various DNA fluorescently tagged, and then let them bind to the hydrogel, diffuse and bind, and then we wash the patch with buffer for quite a few times to remove any unbound target. And then we image the patch under a fluorescent scanner, which is what we've done there. Good signal. This is the patch image with a kind of DNA 210, and then with DNA 141 as a non-specific, and you can see we can achieve very good sequence-specific capturing. It is concentration dependent, so you can actually detect the more DNA you have in solution, the higher the signal on the patch. That's great. So this was in vitro. Can we actually go something closer to something which is more of a final application? Can we actually do this on skin? So you can't really tell on this. So this is the microneedle there on the top. This is a skin sample on the bottom. What we did there, we soaked up, we spiked in the skin sample, if you want, with our target, also labeled with a fluorescent dye, then applied the patch on it, removed it, and tried to see what was captured. So ex vivo testing, if you want, in some ways. We've been able to do this with kind of single dye, again, show very, very good level of kind of specific capturing, and then a dual color system as well. This experiment showed you we could actually kind of penetrate human skin and also detect spiked in targets. But obviously, in an in vivo scenario, your microRNAs aren't kind of fluorescently labeled. So we try to go for something which is more realistic. How do we go from this to an actual sensor? So we've done this experiment by trying to detect unlabeled kind of DNA target, either on chip or off chip. So for the on-chip version, again, same principle. You have your patch with your hydrogel with your PNA attached, put it in solution that contains your DNA target, then remove it and wash it off, and then trying to image the PNA-DNA complex, which has been formed, hopefully, by a capturing of your biomarker. For the on-chip one, we then use kind of fiber-safe as a kind of a double-stranded kind of DNA or PNA-DNA, in our case, kind of a probe. Then you would expect the patch to kind of lit up when the fiber grain intercalates between the base pairs. And then another one, which is basically using the photoclavable linker in here, irradiating the patch, cleaving the bond between the PNA and the hydrogel, eluting the kind of DNA complexes, and then detecting what has been released from the patch. So we've done both with kind of good level of success, not amazing. I think the detection is clearly something that we have to work on. So we know the capturing works well, but detection is still not optimal. So just the next couple of slides just to finish on that and how this project is now evolving. Always very happy. It's very, very preliminary, and some of us have barely started, but this is my first kind of time in kind of a number of years doing academic research to actually put some device on actual live animal. So getting quite excited about that. So very early, no many conclusions to draw from that, but I still wanted to kind of share it with you. So trying to kind of see whether this works. So we have two different mouse models. One, a melanoma mouse model, where in this case, we inoculate the mice with kind of melanoma cancer cells. So use a syringe under the skin, inject the kind of the cancer cells, let them develop. The tumor will develop under the skin, and then we can apply our patch near the kind of a lesion that's been formed, removing it, and try to kind of analyze the microRNA in skin interstitial fluid. Idea in here is to try to see if the same level of deregulation are observed in skin ISF compared to blood plasma. Another model is the kind of cutaneous squamous cell carcinoma using a more kind of spontaneous kind of arising cancer models. We basically kind of exposing the mice to a range of different carcinogens. So that can be kind of chemicals rubbed onto the skin or just some kind of UV radiation, and then wait for weeks for the cancer to start developing. So this is a more realistic model. Obviously, you don't inject melanoma under the skin. In here, you let the cancer kind of forming and progressing. And that allows you, because the patch obviously minimally invasive, to kind of do some point measurements throughout the time the cancer develops. That will tell us how early we can actually detect the skin cancer, and also kind of where we actually need to apply the patch compared to the lesion. What we've just started doing is just that, and that's going to be one of my last slides, I believe, is kind of using kind of now patches coated with hydrogel without DNA on. So what we try to do there is using our patches just to sample ISF and do RT-qPCR analysis. We've done that on four mice, two healthy and two with inflamed skin, so basically mice which just been rubbed with some kind of chemicals. It's not cancer, but it's just to kind of inflame the skin. Then we applied our patch for 20 minutes on sedated mice, then remove them, denature the hydrogel, and that's the cross-linking is through calcium ions, add EDTA, breaks the bond, then wash off all of the kind of liquid. Then we do a total microRNA extraction, normal kind of extraction kit commercially available, and then analyze specific microRNA by RT-qPCR. And if you can imagine, the patch is very small, mice are very small. Weren't too sure whether we could even kind of sample anything or detect enough biomarkers from these small samples. And we are very happy. We've done this with two microRNAs, miR205 and miR21. And we could actually first happy to actually see some kind of microRNA concentration in our sample, which wasn't a given, and even see some kind of pretty large differences in kind of relative concentration of these two microRNAs between the mice with inflamed skin compared to the mice with normal healthy skin. So that's the conclusion, pretty happy pretty early on, and we're going to kind of carry on that. Just to finish, just to insist that everything we're doing is to develop platform technologies, which we can apply for the diagnosis of DNA or RNA biomarkers. All of the work we've done so far is focusing on skin cancer, but we've also started different projects using the same technology for other skin diseases, scleroderma being one of them, in collaboration with clinicians based in Leeds, but also kind of more kind of parasitic diseases, such as cutaneous leishmaniasis, and we're also trying to do that in collaboration between colleagues at Imperial and Cambridge. And just to kind of thank kind of you for your attention, the sponsors, Cancer Research UK, the Leo Foundation is just funding our work on kind of scleroderma, sorry, and scarf for the cancer work. People who did the work. So this project was initiated by a PhD student, Dana, who is now kind of a PI at kind of Coast in a UEA. So very kind of fast trajectory, which is very nice. And Connor was taken over, and he's just 1 year into his PhD, and our collaborators at MIT and ICL. Thank you.